Ladies and gentlemen, welcome, and thank you for braving the cold outside, which I think some people haven't done. So I'm Philippa Saunders, and I'm the Dean of Postgraduate Research for the College of Medicine and Veterinary Medicine. So it's my great personal pleasure this evening to introduce Professor David Hume, who's going to give us the lecture. And David, as you either will know or you're about to find out, is actually from Australia. And during his research career, he has published more than 350 papers. And they've made a huge contribution to science, and particularly to our understanding of one of the most important cells in our body, the macrophage. Now, I know also he takes a very keen interest in developing future science leaders and in training. And he tells me he's just graduated his 45th PhD student, which is a very considerable achievement. So David's worked in Australia, in Germany, in the United States, and currently he's based here in Edinburgh. And like many of the best scientists, he's forged very strong collaborative links around the world. And in 2012, he was awarded the Omics Award by the Riken Genomic or Genome Organized Research Center in Japan. And he became the first non-Japanese recipient of this award. He's received a lot of other awards, which I'm not going to go into, and has also been honoured by appointment as a Fellow of both the Royal Society of Edinburgh and the Academy of Medical Sciences. So naturally, when David moved to Edinburgh in 2007, the research community here were particularly delighted when he accepted the job as director of the Roslin Institute. And it'd be fair to say that under his leadership, this world-leading National Institute of Bioscience has really flourished and gone from strength to strength. So when he joined the Institute in 2007, there were about 200 staff. There are now 500 staff. And he's overseen the move into a £60 million state-of-the-art building at our Easterbosch campus. And the research he's leading at Roslin is focused on improving animal health and welfare, benefiting the environment, developing countries, farmers and the public. And their key research goals include conservation of genetic diversity, which I think is pertinent to today's talk, control of animal diseases, development of new diagnostics and therapeutics for animals, humans, and improvement of food quality and safety. So I think with this introduction, I can't think of anybody more suited to provide us with what I hope is going to be a very stimulating talk on the subject of genes in our food. And I am personally very much looking forward to this talk and hearing the many interesting questions I'm sure you'll be able to pose at the end. So, Professor David Hume.
Thanks, Philippa. So the title of today's talk actually derives from a cartoon of a woman walking into a shop and saying, "I want some food that doesn't have genes in it." And of course, there's a great deal of misinformation about the food that we eat. As you can tell, as Philippa foreshadowed, my accent doesn't come from this side of the world. And when I first came here, I was often asked why on earth I had moved to Scotland. And my standard answer with my faux Scottish accent was, global warming, it's the place to be. Is that okay? So, in fact, the real reason why I came here was, of course, that they had already built a statue of me in the Royal Mile. And so, in fact, this illustrious gentleman who sits on the wall of Old College and this less illustrious gentleman actually share an ancestor, Sir Patrick Hume, the leading figure of the Reformation. But David Hume is actually a hero, as far as I'm concerned, because he is the first great empiricist, a great skeptic, but he obviously also had an interest in genetics and life because he looked with wonder at the immense profusion of beings animated and organized, sensible and active. The whole presents nothing but the idea of a blind nature impregnated by a great vivifying principle. He wasn't a man of few words. But he clearly noted that at least some of these animals came out with various kinds of mutations that we actually not very long after started to look at and say, well, that's actually an interesting animal. Maybe we'll keep that animal and decide to breed from it. So in this evening's talk, I'm going to talk a little bit first about the Roslin Institute and what we do, and why you should know that we are out under the Pentland Hills. And then I'm going to talk about what some might view as the Brave New World, and I think of as an inevitable response to the challenges that face us in this world. So the nature of the problem is fairly well stated by John Beddington, the former government chief science advisor. It's clear that science and research to increase the efficiency of agricultural production per unit of land is critical. There is no doubt that the population of the world is going to keep increasing for a while yet, and the size of the Earth is not going to increase in in the near future. So we need to produce more food with the available land. What you may not know quite as obviously is the strength of animal sciences research in Scotland. And in fact, when I came to Roslin, part of the reason that I took the job was the strength of research in Scotland in the area, and also the opportunities to work with animals other than mice. I have actually cured cancer in mice many times. There is no market for this cure. So it was interesting, and it was like being a kid in a toy shop to suddenly get involved in all of this different kinds of research. But we do have an extraordinary depth, and it's not just me saying it. It's nice that we have Scottish enterprise, completely unbiased, of course, looking at the opportunities for animal sciences research in Scotland and recognising that we have the largest concentration of animal sciences related expertise anywhere in Europe. I think arguably we are already the world leaders in animal sciences. We're certainly the world leaders in brand recognition, because when I was actually taking the taxi to the airport to come here, my taxi driver said, "Where you going, mate?" And I said, "Oh, I'm going to Edinburgh." And he said, "Oh, what you doing?" And I said, "I'm going to work at the Roslin Institute." And he said, "Oh, Dolly the sheep." That's sort of brand recognition you can't buy. So what do we do at the Roslin Institute? So you've all heard of Dolly the sheep, presumably, unless you've been in a time warp. But in fact, what Roslin is really known for in the science world is the science of quantitative genetics. In fact, Edinburgh has an amazing history in genetics, from Waddington and Fisher. We started off at the Roslin Institute as the Institute of Genetics of the University of Edinburgh in 1919. We left the university, as most government-funded organisations do. I think we probably went through something like 11 or 12 different acronyms, the most recent being the Poultry Research Organisation and the Animal Breeding Research Organisation. And all through that period, the people at Roslin were developing different ways of understanding the inheritance of important traits and working out how to do breeding in a more sustainable and rational way. The critical thing, really one of the most crucial things, was the recognition of inbreeding suppression. In other words, that if you just keep brother-sister mating indefinitely, things are going to go wrong, and ways that you could avoid that. The division of immunity, this is actually out of date. I should have changed this slide. The head of that is now Pete Kaiser. We work on many of the endemic diseases that are the major constraint on production, and particularly looking at ways of preventing them and also breeding for disease resistance. One of our major areas there recently has been the recognition that resistance to tuberculosis is heritable. So it's potentially possible to breed cattle that are considerably more resistant than the average Holstein Friesian to tuberculosis. And that would be very good news for cattle, very good news for farmers, and exceptionally good news for badgers. So the next division is the neuropathogenesis division. That grew out of work on mad cow disease and the Creutzfeldt-Jakob disease. These are not only still with us, although in fact I was unfortunate enough to live in this country in the early 1980s and consumed meat although I couldn't afford to buy very much beef at the time. We lived mainly on New Zealand lamb. I nevertheless am prohibited from donating blood in Australia for all time because of that residence, because I might be a carrier. In fact, probably a quarter of a person in this room would be a potential carrier of TSEs, transmissible spongiform encephalopathies. But these are also wonderful models for the diseases that at least a third of us will suffer later in life, that is dementia, loss of cognitive function, because many of the biologies of these diseases are the same. The division of developmental biology is the legacy of Dolly the sheep in some respects, but we look at the development of animals to understand partly how we can improve them, also partly to understand how we can improve their welfare from the animal's viewpoint. And finally, Roslin is intimately associated with the leading vet school in the UK. That will undoubtedly be confirmed in the latest research assessment exercise. And every day of the week, we have companion animals coming through our clinics that have all the same diseases as us. So just as we are living to a riper old age with improved nutrition, so are our cats and our dogs and our horses. And in fact, cats and dogs also suffer cognitive loss with age. Even sheep suffer cognitive loss with age. And I once said that to someone at the Royal Highland Show, and I was asked, how can you tell? Actually, sheep are quite clever creatures, and one of the things as they get older is they lose the fear of dogs, I'm told. So we all overcome our fears with age, even sheep. So why do we do what we do? Well, the obvious thing is livestock improvement, improving productivity. We also work on both livestock and companion animal health. We have extensive interests in biotechnology applications, and one of the projects that I'm involved in myself is to make pharmaceuticals in hens' eggs, pharmaceutical proteins. So we can have the farm as the punchline. And finally, of course, we share many diseases with our companion animals and our livestock. Sometimes we share them directly. For example, in India, both humans and cattle are infected with both bovine tuberculosis and human tuberculosis and are happily exchanging those organisms. So when we talk about One Health, we're talking also about organisms that we share as well as problems. So what we do makes a difference in the world. As I will tell the Westminster government, we generate 12 to 13 pounds of wealth to the UK per annum for every pound that's invested in us. We get lots of commercial outcomes. One of the ones that I think perhaps many of you wouldn't think about is the sexing of chickens. Now this is a talent. It's a talent that the UK is short of because it's a talent that is most highly developed in people from Korea for not obvious reasons, and we have great trouble getting permission to get potential migrant workers to come in and work in the chicken sexing industry. I mean, you know, the Border Authority doesn't see this as an entirely skilled trade, but if you spend more than 10 seconds determining that a chicken is male or female, you are wasting time. So this is an important skill. We've developed a technology which can enable us to sex an egg based on the amount of material that is on the end of a needle used for in ovo vaccination. And it can be done with a manifold of 1,000 eggs at a time. Now, female chickens are not very good for meat production because they consume more food per unit meat. And male birds are obviously not entirely useful for the production of eggs. So it's desirable that we identify the male and female eggs, and the ones that we don't want go into omelettes rather than going on to progeny. So these sorts of things make a fairly serious difference. Roslin Institute is this wonderful building out just underneath the Pentland Hills. We have grand plans for further development of the site. We are building 2 large avian facilities to be the National Avian Research Facility, and we have an ambition to build a large animal research centre that is specifically directed towards improving animal health and welfare. So that's the sales pitch, if you like, for why Roslin is something that you should be proud of in your local community. It is probably the best known brand in animal sciences and one of the best known brands in science generally. But what Roslin has been involved in is something that you may not have actually been aware of at all, and that is what one might call the livestock green revolution. In the past 20 to 30 years, the average productivity of our livestock has massively improved. So, for example, in the pig industry, we have 50% more piglets weaned per sow per year. They have, on average, a substantially increased lean meat percentage. They have a greatly improved food conversion ratio. That's essentially the ratio of food out to food in, which is the best performance measure going. And that's also effectively taking all of those things together. We have almost doubling in the amount of lean meat produced for the amount of food given to pigs. Similarly, in the chicken business, the breeding of broiler chickens has reached the stage where they reach 2 kilograms, which is the normal slaughter weight, in 40 days instead of 100 days. Their food conversion ratio also improved to the same extent. Similarly, with laying birds, the 80% increase in the amount of eggs per tonne of feed, increase in the number of eggs per annum, and finally the dairy cow, which is a metabolic athlete. This is an animal that massively efficiently converts food into milk. Now, some people are disquieted by this, of animals essentially as production units. But the reality is that there isn't really a choice. And the argument that we could all become vegetarians is also not a viable argument, because high protein plant food cannot be produced on most arable land at the level that would be needed to provide protein nutrition to the world's population. So it's simply not an option. In terms of the effect of animal protein where we have protein malnutrition, it's believed that a single egg per week for a child in Africa is the difference between normal cognitive development and poor cognitive development due to protein malnutrition. So most of our current consumption of animal protein really is dependent on the improvements of productivity. Improvements of productivity have a secondary effect. If we consume less plant material to produce the same amount of animal material, then per unit production we produce less methane. We produce slightly more ammonia in broiler chickens, considerably less nitrous oxide, and so the global warming potential index is improved for all of these animals. The animals that haven't really improved in the last 20 years are beef cattle and sheep, the ruminants. That is largely true in the UK. Of course, it isn't true in countries that don't have the common agriculture policy, where there's actually serious competition for production. So there speaks the Australian. And in Scotland, rather amusingly, when we go to farmers and show them the wonderful Texel ram, they will look at it and say, that's an ugly looking beast. And we can tell them that they will get £5 more for every lamb from this ram as the donor of semen, and they're really not interested. So we do have a way to go with improving the productivity and reducing the global warming potential of the ruminant industries. So the genetic real world. Firstly, every single thing we eat is genetically modified. if it weren't, human population growth could not have happened. It has happened because of genetic mutation, as a direct consequence of our ability to produce more food, and it continues to be dependent on it. If we talk about the genetic real world now, billions of acres of transgenic plants are under cultivation worldwide. Even humans are arguably genetically modified. The antibiotic revolution has already, and we can actually tell in archive human genetics versus current genetics, that the antibiotic revolution has enabled people to survive who would not have survived to bear children prior to the onset of antibiotics. So we are modifying our world so that what might be called mutants, us, can continue to live. Companion animals are, many of them, mutants. I had a conversation some years ago about research that was going on at Roslin with a woman who breeds pedigree dogs and was criticizing us for creating animal models of blindness. And I said, well, the ethics is quite straightforward. You breed mutant animals that are severely genetically impacted solely for aesthetic purposes, and we do them to save the sight of children. So which of them is ethically justifiable? And finally, when we talk about the genetic real world, we don't really take account of the major impact. We talk about releasing genetically modified organisms as a threat. The greatest threat of all is moving organisms from one environment to another in which they did not evolve. And feral animals and plants and microorganisms are a much greater threat to environments than anything that we could create genetically modified. In Australia, again, the entire landscape has been shaped by feral rabbits, feral foxes, feral goats, feral donkeys, feral camels, feral pigs, none of which existed prior to white settlement, with the one exception of the feral dog, otherwise known as the dingo. The real genetically modified crops that in Europe, we have the minor insanity that we will not allow so-called GM crops to be planted. But the real GM crops are the ones that we all eat. And this database shows 2,500 mutant varieties of wheat, rice, and maize. Note how they were created, developed through physical and chemical mutagenesis. So this is treating the plants with harmful chemicals or radiation, breaking up the genome, selecting the things we want with no idea what else we've done in the process. But that is not genetically modified. So those plants are being planted all over Europe. No more predictable what they're likely to do than the transgenic plants we talk about. It's worthwhile remembering that the first genetically modified plants were actually made in response to the publication just over 50 years ago. And in fact, I read it not long after it was published, of Silent Spring by Rachel Carson, which pointed out the folly of massive distribution of pesticides and herbicides, the obvious iconic loss of the peak predators, the eagles, the raptors, through the accumulation of things like DDT. And that led to the banning of DDT and massive changes in the use of pesticides and herbicides. But as Maurice Maloney, who talked here a few months ago, was actually working with Calgene and was making the point that when he started working on making genetic modifications of plants, his motivation was to essentially reduce the use of pesticides and somehow maintain the productivity that had been created by the Green Revolution and was now being threatened with the environmental consequences of widespread pesticide and herbicide use. So the full incomprehensibility of some of the choices is illustrated by the choices that one has in canola. So at the top we have the product of the evil Monsanto. So Monsanto engineered canola to have resistant to the herbicide glyphosate, and in response to the concerns of the environmentalists who were basically no GM, they created sterile seed. And then, of course, they sold the seed, and being evil companies, of course, they made money. However, it was not their intention when that technology was made to create sterile seed. And so now that they sell seed, the company is essentially criticized, and they actually bought the technology. The alternative is the clearfield varieties, which are resistant to the imidazolinones. These clearfield varieties, and it's not only canola, it's actually in quite a number of other crops, are actually created by chemical mutagenesis, and then they're selected for the loss of the enzyme ALS, which is required for the synthesis of branched chain amino acids. This is an enzyme that doesn't exist in humans, so we don't have a problem. We get our branched chain amino acids having had them made for us by plants. These plants are not GM by definition. And so the choice is made in Western Australia, where canola is grown extensively, that the Roundup Ready crops are not used, or they are to some extent. These are heavily used. Glyphosate has a half-life of about 24 hours. Imidazolinones have half-lives of weeks to months, depending on rainfall, and they accumulate in soil. So which is the more desirable technology? So we turn to animals, and the unfortunate company AquaBounty, which is, I think, almost there in the United States to get approval to release the AquaBounty salmon. I won't go into the details, but essentially what this is a transgenic fish which has a growth hormone gene from a fish that grows at colder temperatures, and so it can start to grow at colder temperatures. It reaches commercial weight almost 7 times more rapidly. The only production animals are sterile females. There is absolutely no possibility of escape from the barrier to sterility. Nevertheless, the opposition to this technology puts the view that these animals will compete in the wild, and therefore there is a threat of release. This, on the other hand, is something that was legal to import into the United States until quite recently. It looks considerably less threatening in a fish tank, but if it gets into the Great Lakes, it will completely denude them of all other fish. It has no known predators. It's rather evil looking, and it can actually migrate across land between bodies of water. So this thing, to me, is an awful lot greater threat than this. The other casualty, if you like, of opposition to genetic modification in animals is the so-called EnviroPig. Almost as soon as these were published, their existence, we had the songsters coming out with a song about the wonderful EnviroPig. So the EnviroPig was perhaps a little tenuous, the name, but you maybe can't read this, but the enzyme phytase is required to break down undigestible phytate, which contains phosphate in the diet, and is a way of basically the way that the pig, and for that matter chickens, which eat a lot of grain, gets phosphate out of its diet. Now, neither pigs nor chickens have phytase, So they digest the phytate very inefficiently in their terminal bowel, and that's how they get phosphate nutrition. So the very bright idea was to express phytase as a transgene expressed only in the salivary gland. So the animal then actually digests the phytate and releases phosphate and does not require phosphate supplementation in the food. That in turn means that there's considerably less phosphate in the poo, which is considerably less contamination of the environment. So that seemed like a good idea. The argument was put quite early on that these animals would somehow be compromised, and in fact they took the company that made them actually put them under selection for increased productivity, and the transgenic animals actually diverged and performed better under selection than the animals from which they were derived, and actually had a reduction in the average age to reach production maturity. So the interesting thing is that it was possible to genetically select for even better use of the transgene. So unfortunately, perhaps for the environment, the animals were not approved. The company went under. And the alternative to this approach of transgenes is actually to feed phytase in the feed. So the phytase is made as a recombinant protein in bacteria. So it's a genetically modified protein by any reasonable description. It's fed. It costs considerable amount of money to make it. And it's nowhere near as efficient as expressing the transgene. So there is still significant phosphate in the environment as a consequence. So if we don't resort to genetic modification, of course, nature does it for us. Every individual in this room is a mutant. Don't take it personally. You're all carrying somewhere between 100 and 200 serious genetic diseases. As long as you don't choose to procreate with your siblings, this is probably not a serious problem. But of course, some of you have desirable mutations as well, and those sorts of mutations arise in every generation. So if we don't introduce genetic modification, and we don't do what we can in fact do in mice, but has not been done seriously in production animals, we don't deliberately irradiate them or feed them nasty chemicals to get mutations, nevertheless we can select animals to breed based on shared desirable traits, and sometimes we end up with predicted outcomes. But things have got an awful lot more predictive, especially in animals such as dairy cattle, where we have a single bull sitting in glorious isolation producing semen. I should say, incidentally, with respect to tuberculosis, in the 1930s, when we had tuberculosis-infected cattle, we just killed them, and that broke the epidemic quickly. Nowadays, of course, the bulls never get exposed. So the bulls sit producing semen, and the poor old cows out in the field getting tuberculosis, and as fast as a tuberculosis-infected cow is culled, we bring in semen from a bull who was never tested. So we're no longer selecting in favor of tuberculosis resistance. But as you can see, this is the ancestral Friesian. This is a modern Holstein Friesian bull. What we can do now is actually assess the genetic barcode literally at hundreds of thousands of positions in the genome, and we can identify the best match between that barcode and a whole set of traits simultaneously. So this really is computational breeding. We assess what we call an estimated breeding value for any of those traits, and then we decide that this bull should be used with this set of cows to get this desirable outcome. The application of that in the UK is part of the National Dairy Fertility Index, and one of the consequences of breeding cows that produce so much milk is that their fertility declines. Now, everybody who knows anything about biology knows about lactation suppression, so that's a natural biology, and there was a necessity to overcome that to some extent to maintain cows in production on an annual cycle. So with this approach, literally in a matter of a small number of years, we are turning the corner on dairy fertility, finding a sustainable compromise between total production and coming back into cycle and producing milk again in a subsequent year. But the astonishing thing about this technology is that we can measure all of these things in the progeny of a bull, and we can say to a particular farmer, what are the most important traits for you where you live? We may not bother specifically breeding for tuberculosis resistance in Scotland, where we don't yet have it. And we might, on the other hand, breed specifically for calving ease, because we might have animals that are going to have unsupervised calving. The other thing that when we talk about animal biology and we talk about production animals, we tend to anthropomorphize. One of the best examples of this is the wonderful free-range egg. Now, when I buy eggs in Sainsbury's, I go to El Cheapo cage eggs quite deliberately, because I've been down to the free-range facilities not that far from here, and the current legislative framework for free range is a big shed where the animals are okay to run around, 75,000 in a shed. Open up the side of the shed and the animals are free to range. Now, chickens get predated from above and they get predated from below. The last thing a chicken wants to do is run out in the open air. They are forest animals originally, so if we had a free-range forest, that would be fine. But they get chased out. So the bottom of the pecking order gets chased out. They in turn pick up pathogens from the field, bring them in. The sheds can't be fumigated, so we have red mite throughout the sheds. So it's debatable whether this is optimal from a welfare viewpoint. It's also debatable whether it's desirable to suddenly put birds into a free-range environment, because the other alternative is the enriched cage. So if you go into a facility where there are wonderful enriched cages with 12 birds per cage, a nice little nesting place where the birds can go, and you walk down that row of 180,000 birds in a shed in their enriched cages, then in every cage there is a single fully fledged bird, because we haven't selected against pecking behaviour. So the birds that we currently have been adapted and bred for life in a cage. If we want to change that, then we need to think about breeding for behaviours, and that is also possible. So many of the natural behaviours, we nurture the illusion that romping across the countryside is the best thing for a cow. And again, there was a proposal to build an 8,000 cow shed in Lincolnshire with automated milking and everything, and the major opposition to that, other than pollution, which was a justified opposition, was that this was unnatural and the cows would be unhappy. On objective measures, we can measure stress in animals. If the animal is stressed, it is less productive. So there are objective measures, but much of the objection to production systems that we need to think about is based on anthropomorphic views of behaviour and welfare. So what is possible, what is desirable in manipulation of animals, if you like, for improved production? Well, the first is what we do all the time, and that is heterosis. Routinely, when we breed production animals, we take animals that are as different from each other as possible, having bred them to essentially be genetically compromised in order to be in somehow aesthetically interesting, and we cross them. In doing that, we basically ensure that we only get one copy of the bad genes that have been bred into one of the donors of that hybrid. So hybrid vigor is a well-known phenomenon. It's also used extensively in plants. When we talk about plants, of course, our favorite hybrid is in fact wheat. Wheat is a hybrid of 3 different species, 2 of which came together in the wild. The third was actually introgressed deliberately, and we don't know what it was. If that was done now, there would be every reason to ban it because it's quite clearly genetically modified, an interspecific hybrid. It's also quite clearly dangerous because 1% of the population has celiac disease, so clearly causes allergy. So I'm not going to advocate the banning of wheat, but it would be just as rational as banning any other genetically modified foodstuff. So we use hybrids routinely, but we get into funny territory when we talk about hybrids of animals. They are possible. It's possible to make geep, which are sheep-goat hybrids. It's possible and in fact is done routinely to make chorizo, which are llama-alpaca hybrids which have very desirable wool. Cloning, of course, is the legacy of Dolly the sheep. It's done all over the world, and it's done with all kinds of species, cats, dogs. The most famous cutting horse in the United States, something called Blue, was cloned to produce Blue 2. And Blue 2 is used solely as a broodmare, so has never actually gone out and shown her stuff, and her foals are something worth some extreme amount of money in the 6 figures. So this is obviously lucrative, but you will have many of you will remember the press about the importation of cloned cows from the United States. That also led to a wonderful cartoon of a woman sniffing a bottle and saying, "Does this milk smell cloned to you?" Mutagenesis, as I said, we haven't done that using chemical mutagenesis in animals. It happens naturally with quite reasonable frequency. We're now in a position where we can sequence the entire genome of animals routinely. And so rather than looking for phenotypes, if you like, the traits that we might select, we'd actually sequence the entire genome and say, we would like to have that point mutation, and we're actually working in that space with some of the major breeding companies. From their viewpoint, they would like to get rid of the undesirable mutations. From our viewpoint, we would like to actually cross them so that we can create animals that lose particular genes and essentially see what those genes do. I'm going to come back to targeted mutagenesis, which is the final technology I want to talk about. So cisgenesis is a technology where you take a desirable gene from a breed of the same species or closely related. We already talked about taking a gene from the Chinook salmon and putting it into the Atlantic salmon. That's technically relatively straightforward and has been done. In some cases, it would be a quick fix. For example, in Charolais cattle or Texel sheep, they have a mutation that causes double muscling. That's part of the reason why Texels and Texels in particular are so ugly. They've got this massive back end due to double muscling, which is a single mutation. We could introgress that by just conventional breeding, back crossing to the Texel, from putting it into a different genetic background and then mating back in conventional breeding, but that would take 5 to 10 years. We can achieve the same thing by simply moving the piece of DNA from one animal to another. The one that we actually tend to call transgenesis is the moving of genes from an unrelated species. It's already possible to create cattle, and it has been done, which are making natural antibiotics. So there were cattle made nearly 10 years ago that express a protein called streptolysin in milk, which essentially sterilized the milk, largely rendered pasteurization unnecessary, but also massively reduced the incidence of mastitis, which is infection of the udder, which is a major welfare problem for the cow. So that, of course, was something that didn't come to market, but that is something that we might think about again. As we ban antibiotics in feed, and they're routinely put into food, into animal feed to essentially get a growth advantage, and to be honest, we don't actually know why, because they're at sub-therapeutic concentrations. We're going to need a substitute. The amusing thing again in terms of public policy, the elimination of antibiotics from food has actually, routine feeding has actually increased the use of antibiotics in livestock production, because instead of having low levels prophylactic, they now have high levels therapeutic to deal with the periodic infections that go through every production facility. So from the point of view of the animal health companies, the banning, paradoxically, the banning of antibiotics was a good thing. So pick the transgenic chicken. So one of these animals is a transgene. These are silkies. They have a mutation that was selected. These animals would not do terribly well in the wild because they can't fly and they have very odd feathers. But they have this melanotic pigment which is particularly favoured in Chinese. So this is the Chinese silkie. This is our local icon, the Scottish dumpy. Scottish dumpy can't be mated to each other, or you lose a quarter of the eggs because the 2 copies of the gene that causes this is lethal, the same as the Manx cat. This is the bare-necked chicken, which actually does very well in hot climates because it loses heat through the neck. We in Roslin, Dennis Heaton and his group found exactly what kind of mutation this is. It's actually a great big slab of DNA that has moved from one part of the genome to another. So pretty much as disruptive as any transgene. This, of course, is the featherless chicken. Saves money on plucking. Doesn't do all that well, so it's not a particularly productive bird, but nevertheless possible. And this is the transgenic chicken made by Helen Sang's group in Roslin that does not transmit avian flu, so is potentially one of the possible defences against an avian flu epidemic. Ooh. So the technology that's coming is what we call genome editing. We now have a technology where it's possible to actually take a single letter, a single base out of the genome, or to substitute it with another base. And my colleague Bruce Whitelaw in the Roslin Institute headed a team that made the successor to Dolly, Pig 26. We haven't thought of a better name for it. Any ideas I'm happy to hear. So this is this animal has a single point mutation in a gene that we believe would confer resistance to African swine fever. The rationale behind this is that warthogs are resistant to African swine fever and domestic pigs are not. African swine fever is, according to this site, there has never been an outbreak in the UK, and the incidence is very low in the rest of Europe, although there have been outbreaks in Russia and in Africa. The last time I saw, there was no barrier between Russia and the rest of Europe, and this, of course, is the kind of misinformation that we get in the face of any technology. But this is the actual technology is, if one is open about it, we could make that mutation and just put the animals out in the shed, and if you said, how did that animal come to being, I would say, it wasn't in me. I didn't do it. It could have arisen naturally. So this technology is already being applied in plants. So there are genetically modified plants, and they're genetically modified in a completely different way from the kinds of modifications we've made in the past with chemicals and radiation, etc., where we had no idea what we were doing. Saturation bomb, select the plants that are lucky enough to survive, and then breed from them to get the variation that we want with no idea what else we've done. So genome editing is a technology where, regardless of the rights and wrongs of whether you think it's right to do it, at least we have some idea what we're doing. So this, of course, is everyone's. This is a wonderful sculpture, a hybrid of what looks like about 3 different species, and it's everyone's view of genetic engineering in animals, the brave new evil world. To return to my earlier point, every single thing we eat is genetically modified. There are no foodstuffs that we eat that have not been subject to selection by humans, even if that selection is only choosing which animals to mate based on a desired outcome, or which plants to hybridize based on a desired outcome. We have done things that did not happen without human intervention, and so we have modified their genomes, we have modified the genomes of the world. We are the greatest force of evolution the world has ever seen. So what we have to ask ourselves is what can be justified in the future, assuming the technology is possible. Is it justifiable to select for disease resistance? Is it justifiable to modify for disease resistance? Is it justifiable to make transgenic chickens that are resistant to avian flu if the choice is, as happened in the last avian flu outbreak in Southeast Asia, the choice is to lose literally millions of chickens? Or in Europe, as is almost inevitable, the choice is to have a widespread epidemic of African swine fever. So it's a question that I think we all need to ask. Can we ever have a sensible cost-benefit risk analysis? I am not advocating the unfettered use of genetic modification. There are many things that are done routinely that I think are environmentally unsustainable. But what we do need to have is some kind of sensible cost-benefit risk analysis, because the choice is not do nothing. The choice is starve or do something. Widespread world starvation or do something. So we do need to ask, what are the compromises? And I think the question one has to ask in this respect is, what would you eat? What would you be prepared to eat? I personally would have no trouble eating fluorescent chickens and fluorescent ham. So Dr. Seuss foreshadowed this. Sam didn't like green eggs and ham. I would be perfectly comfortable with it. It's just food, it's just protein. Secondary question, what if you were starving? So then your choices become rather more compelling. So thank you for your attention, and I'm happy to take questions.
So at the end, you kind of made the point, at least what it seemed to me, as if this issue of food scarcity and world hunger is a black and white kind of issue. Like you have a choice to make, and we can either starve or resort to genetically modified organisms. And I have a question for you. I wonder to what extent do the researchers at the Roslin Institute, and I guess yourself and the people you work with, take into consideration the idea that world hunger is caused by many factors, and that starving kid from Africa that you mentioned a few slides ago, the starving kid in Africa does not need a handful of wheat. That starving kid in Africa is most likely starving because the people who are producing the food there do not have food sovereignty. And I think there's, I don't know, I guess there's a larger political and socioeconomic situation surrounding food, like world hunger. And I'm wondering, like I said, to what extent is that taken into account when you're pushing for GMOs in third world countries?
So I'm not necessarily pushing for GMOs. I'm pushing for rational debate. So the opposition to genetic modification is irrational. And I go back to my illustrious antecedent who was the skeptic. And philosophical inspiration, let's see the evidence. Most of the opposition to genetic modification per se is focused on a single technology with an arbitrary definition which is not sustainable. So the question of the politics of world hunger is of course well taken, and we participate in all sorts of approaches to the solution of sustainable improvements in productivity delivered locally. So I actually am on the scientific advisory board of the National Institute for Animal Biotech in India, and we're working on a project to breed animals by conventional breeding to improve the resistance of buffalo to tuberculosis. So that's not genetic modification as you would define it, or as perhaps not you, but perhaps the public would define it. It is nevertheless genetic modification. It is selecting which animals would breed. My choice, not the animals. So that's really what I'm getting at, is that there isn't a boundary where this side is genetic modification and this side is not. And that's what I object to most. It's to look at individual technologies on their merits and say cost, benefit. So the solution to world hunger undoubtedly is a much more complex issue, but I'm also an advocate of the think globally but act locally. The fact that there are lots of other reasons does not mean that we should not try to contribute in our way to offering sustainable solutions.
Hi. I took a little bit of objection to some things, especially. Well,
I was meaning to be provocative.
Yeah, no,
so
you said
if
you
you jumped, that's cool.
No, I'm a biology student, and I've just written an essay about GM. So I've done quite a lot of research recently. But actually, one of the things that offended me more was a disregard for the idea of vegetarianism being a good option. Like, obviously, that there is quite a lot of land that is suitable for things like sheep and not suitable for crops.
No, so that kind of misses the point. So high protein crops require high quality land. So I'm not saying that vegetarianism is unsustainable. I'm saying that vegetable sources of protein are not sustainable in every part of the world, because they simply do not have.
But in the UK, we could be more vegetarian. We should reduce our carbon footprint.
Eat less meat.
Yeah, we should eat less meat. So you would advocate people eating less meat?
No, absolutely not. What I'm saying is that a absolutely vegan option is not.
No, I don't advocate an absolutely vegan option. So I advocate a compromise. I advocate a situation where we could actually have a balance of animals and crops, which would be mainly crops. So that would be the most sustainable solution.
Couldn't agree more.
So you do promote people to eat less meat?
Yeah, absolutely. Because
despite promoting increased meat production?
Well, you've still got to, even with the current trajectory, given that meat consumption is rising rapidly worldwide, it's a luxury for us because we have an unlimited protein availability in the Western world, unless we're on the poverty line. But it's not a luxury for other parts of the world.
So we could choose to eat less meat?
Absolutely. Absolutely. And
decrease production of that meat?
No.
Well, if we were eating less meat, we've decreased production of meat.
No, because we're not an island. We are an island, but we're not isolated from the rest of the world. So if we produce more meat more efficiently on our land, we should be exporting it, so that it's not grown on marginal land that would be better used for arable in other parts of the world. So it's a world economy and it's a world problem. So you can't advocate an isolationist approach in the UK that we should produce less meat so that we eat less meat. We should produce more and we should export it.
Sorry, it was just to do with your plant-based protein argument again. It was, what about fungal-derived protein and insect-derived protein?
I think that all solutions, all technology solutions to obtaining protein will be necessary in the future. I mean, I actually like the idea of integrated vertical farms, within cities, so that we can return some land to essentially non-food production. And I think there are all sorts of solutions that are possible, including using fungal sources of protein.
Thanks for a very interesting talk. I work in health, and I was interested in what you were saying about antibiotic resistance in terms of antibiotic supplementation of animals and the subsequent, I'm not sure if I understood you correctly, but were you saying that after removing antibiotic supplements in their low subtherapeutic levels, they're actually requiring higher doses of therapeutic antibiotics? I didn't understand why you would require, if they were subtherapeutic in the first place.
We don't know exactly why antibiotic supplementation improves production, but it does. And presumably, part of it is that it alters the microflora in such a way that the animals are, for reasons we don't understand, although the antibiotics are below therapeutic levels, they nevertheless confer resistance to infections, because the outcome is that the animals get infected more often and acutely symptomatically infected, and so they're actually treated then with high-dose antibiotics. And you're allowed to do that as long as there is a recovery period after the antibiotic administration before the meat enters the food chain. So the consequence of removing antibiotic supplementation is a higher use of antibiotics, which actually drives more resistance.
So I suppose, so having understood that correctly, would you therefore advocate the use of antibiotics supplementation in foods for animals?
I think it's going to come when, well, I think there are other ways of managing it, but one of them is actually to breed, because we've not bred animals for disease resistance, is to actually breed them to have naturally higher levels of baseline resistance. In other words, to breed them to compensate for the absence of antibiotics, because we've had antibiotics in feed for 50 or 60 years, so most of the recent productivity gains have been animals that have been bred to grow with low-level supplementation with antibiotics.
It's not a terribly controversial question, I'm afraid.
Oh, good. I can relax.
My question was more, you mentioned that we are effectively mutants and that we have effectively changed our genetic code. And you mentioned also that there was a difference between the records that we have of the human genome before and after the application of antibiotics. And I was just wondering what those differences were that you mentioned.
There are quite a number. Let me go back to the heritability of infectious disease resistance is very high. So if you have a first-degree relative who had colon cancer, first degree meaning brother, sister, parents, then your chances of getting colon cancer are something like 3 or 4-fold elevated, and you should have routine colonoscopies. If you have a first-degree relative who died of an infectious disease, regardless of what it was, your chances are elevated something like 10-fold. So resistance to infectious disease is very highly heritable. So very rapidly, if you remove the selection pressure of infectious disease, you stop having half the kids dying in infancy. And it's a nasty thought, but what it means is that you enable people to survive who would not previously have survived and to pass on their genes. So the consequence of that is if we run out of the efficacy of antibiotics, we will be in deep trouble. We've had 4 or 5 generations, probably 4 generations of children who have grown up with considerably less threat from infectious disease. So, and it'd be quite quick. Selection can be incredibly quick. One of the examples of human selection in Scotland, the antlers on the deer are getting smaller because tusked elephants in Africa don't have tusks. That's selection. So the same is true of infectious disease.
David, do you think some of the disquiet with this idea of genetic modification is one of speed? So there's natural selection, which we're all aware of. We've had hundreds of years of people crossbreeding animals, and now there's somehow a feel that it's slightly out of control because we're doing it rather more quickly.
It's an interesting question. I think a lot of people feel like we're playing God, and it's much more immediate when we're doing it with animals than when we're doing it with plants. I mean, nobody is particularly concerned about hybridizing an apple tree, which we do all the time, or about hybrid wheat. But when it comes to manipulating animals, we have an even greater concern. But it's actually not new, because in the 19th century, when line breeding, so most of the major breeds of cattle and sheep and pigs and chickens, 19th century, so Darwin got his ideas from animal breeding. And line breeding was preached against from all the pulpits in Scotland. It was evil, brothers and sisters mating, which is what they did. They just mated back and back and back for the desired trait without worrying about inbreeding. So in some respects, the concerns are not that new. Even the concerns about what we can do, it's surprising the sort of double standard, because people who are uncomfortable with that will be sitting by the fire, stroking their King Charles Spaniel, which has got mitral valve disease. That's why they're lapdogs, because the poor little things have got such defective hearts that they can't run around.
Oh, he's a bundle of laughs, isn't he? Let's have another question here.
Hi. I guess to move away from animals for a bit, they're a really interesting topic, but I just wanted to talk to you about seeds for a little bit. And
not quite my area of expertise, but I'll.
Okay. And I noticed that you brought up the big bad evil Monsanto earlier, and I don't associate genetically modified organisms with Monsanto, although they are heavily involved in that field. But there has been a history of the use of genetically modified seeds in agriculture as a way of gaining more power over the food system. So because when you do create these seeds, you obviously have the rights to them, you have a patent over them, and you then can sell them and make money from it. And seeds have been manufactured in different ways according to different traits that they are wanted from them. And one example could have been the seed that would not reproduce more seeds. So farmers would then have to continue to buy more seeds. And that was obviously economically desirable for the companies that manufacture them, but not for the farmers themselves. And in a world today where...
I should emphasize that that was inflicted on the company, sterile seed.
I'm sorry, what? Two people just said this.
So the sterile seed was inflicted on the company for environmental reasons. So that was the only way that they could, the first transgenic crops, it was a requirement that they have sterile seed so that they would not propagate in the wild.
Yeah, but the intention was to make money. They were not doing it for the benefit of the farmers. But more and more farmers are disappearing now, not disappearing out of thin air, but obviously dropping out of their business, unfortunately. And I'm really interested to see how genetically modified seeds nowadays can promote the livelihoods of farmers and help them rather than assert dominance over them and make them dependent on these packages of seeds and pesticides and fertilizers.
So I agree with you. Personally, I have a radical view of patenting. I think it should be abolished. So I think that people should work on trademarks. One of the interesting consequences of the rapid advances of genetic technology is that, in fact, it would be possible for an amateur to do genetic manipulation. So it's becoming relatively low tech. So that's going to change the marketplace quite dramatically. And you know, I would say that in a marketplace where the technology is relatively available, then the competition will be based not on exclusivity but on quality. So if the seed is worth buying and the profit margin is enough, it will be bought. And it'll be the choice of storing seed or buying seed. The buying of seed, so the most widely used potential technology, which is not all that, well, for wheat, is actually hybrid wheat, which is also widely used. And so wheat is self-fertilizing, so you sterilize one recipient of the hybrid and fertilize it with the other, so you generate a hybrid that has desired traits, 15–20% greater productivity. That is becoming really easy to do. So it's impossible to patent hybrid wheat, and you could just get out in the, you know, simply just experiment with varieties that are freely available, hybridize them, create your own private stock of hybrid wheat, and thereafter plant that, and nobody could ask any questions. So I think the technology is moving very quickly to a point where it's also being distributed around the world. Some places don't bother with patents. Yeah, I think the dominance of the big agri companies is fading, and I think that's also true in medicine, human medicine. The big pharmaceutical companies are finding that life is a little more difficult than it used to be when they had sort of global dominance. They've got bigger and bigger and bigger, and then the little guys come through and say, well, we can actually do this really easily, personalized medicine, little niche, and they break up the monopolies. That's the normal cycle of, as my illustrious namesake actually, who was also the first great economic historian, pointed out.
Have any cases come to light so far of unforeseen consequences from GM, you know, despite testing in the lab or some field testing?
The examples of unforeseen consequences, I don't think so. I mean, partly because, well, I think that the major transgenic crops like BT cotton and Roundup Ready, maize, you know, the herbicide resistance, there have been escapes, and there's been unfortunate press about, you know, seed getting into adjacent paddocks and then the companies claiming ownership. So I think that's probably the area that's had the most publicity. I think it fades into complete obscurity compared to the consequences of introducing feral species into new environments, including potential animals and plants to be grown for food production. So I think ferals are a much greater threat because they are not compromised for production. But I don't have an example. Most of the claims that were made, the most infamous being the claim that transgenic potatoes were somehow caused allergy, have been published in newspapers rather than creditable scientific journals.
I'm afraid this is going to be a bit of a basic biology question.
Oh, that's good.
Comparison, but,
I can answer them.
Well,
I think.
When we go down the route of genetic modification in single step rather than slow introgression or other processes that have happened in the past, when we consume an organism that's been modified in that way, depending on what kind of trait has been modified, but can some of that genetic modification end up in us, either directly in our own genome or within our own microflora, microfauna, and so on? Is that possible?
It's not technically completely impossible.
Yeah, thanks.
That's what scientists always say.
No, so there are, when we talk about transgenes, one of the things that we don't think about is that transgenesis is a natural process. Pieces of DNA jump around within our genomes, between genomes. We have, within our blood, we have these tiny little DNA viruses which adapt to us. And every one of us has got 10 to the 7 virus particles per mil of blood. And those things constantly mutate. And we can potentially transmit them to our children, to each other. They can integrate into DNA, so they can invade our genomes. Very, very rarely, they will get through to the germline. And we published recently one of the other things that, in fact, my own laboratory was involved in, is that we have jumping genes that jump in and jump into different places during our life. And it's one of the mechanisms that causes mutations in our brains during our lives, that these pieces of DNA, which we call retrotransposons, can actually go out and go back in somewhere else. But they can also go out and go somewhere else. And they can pick up pieces of sequence in the process. So technically, moving pieces of genomic information around has happened throughout evolution, and it could happen again. But it could also happen if we ate a cow. You know, regardless of genetic modification, we could get an active transposon, fortuitously active at that time, getting into our digestive system, invading our genome, and then being transmitted. It just doesn't happen very often. But it clearly has happened in evolution. The best known example is actually the domestic dog. And the reason dogs are so diverse is when we first domesticated them, somehow we co-selected for a mobile genetic element. And that's the major cause of much of the diversity amongst dog breeds, is this piece of DNA is jumping around. And so the difference between a Great Dane and a Pekingese is mobile pieces of DNA that have inserted and changed the genome.
So although the process is natural and evolutionary, it's not impossible for a patented gene to end up in a human being?
It's not likely, but it's not completely impossible. But it's also possible that we could get it by eating a mushroom that happened to have the same bacterium on it. So the fact that we consumed that piece of DNA as part of a cow or part of wheat or part of a fungus is irrelevant. It's all just DNA.
I just wondered, given that that potential exists, and I take it, I appreciate that it's a small probability, that maybe your argument about comparing it with feral animals being introduced, if that's a lesson that we learned from the past, that we shouldn't rush into introducing feral animals, and or rather, you know, people introduced feral animals for the wrong reasons and didn't think it through. I wondered if you had any reservations yourself about the ethics when you're sort of giving us pause to think about, well, we need to have a rational debate about this. Are there any things on the sort of con side that would make you think, well, these are downsides of GM as an overall approach?
It's all got to be a risk-benefit analysis. There is no such... So the opposition to the use of GM takes an absolutely minuscule risk and does not weigh it against a large potential benefit. If the benefit isn't there, then the technology shouldn't... Then you weigh it up and say, well, the risk is finite, the benefit is not finite, forget it. But what we don't have is rational risk-benefit analysis, because there is no such thing as a no-risk technology. There's absolutely nothing we do. The most dangerous thing we can do is go to bed, because everybody dies in bed. So we should ban sleeping. Yeah, I mean, yeah, there's sensible risk-benefit analysis is what's actually needed.
I just wondered, I appreciate that, but if we were to say that this is a safe space where we can talk about both sides of the argument, what would you say would be the leading risk? Or are you not comfortable talking about that side of things in terms of...
No, I don't actually see that there is a significant risk compared to the benefit, because we eat DNA all the time. Exactly how we eat it, what we eat it from, is utterly irrelevant. Doesn't matter what the vehicle is, all it is 4 letters in a line. You know, in the end, it's just DNA. As I said, I would have no problem eating green fluorescent protein. It's just amino acids. You know, but then my attitude to food, you know, there's two words for eating in most languages: to feed or to eat. I just feed.
Thank you for an interesting talk. What do you think of the disappearance of the British bees? And what type of cancers did you cure in mice?
Well, I've cured melanoma several times in mice. I've actually also cured a Lewis lung carcinoma, which is a nice transplantable tumor that is a great threat to inbred mice. What else have I done? I've done squamous cell carcinoma. So bees. I don't think I have. I think that's one of the few things I don't have an opinion on. The disappearance of disappearance of British bees is a complex phenomenon. Disappearance of bees all over the world is actually, in some measure, associated with monocultures, and so the reduction of diversity of habitat. And that's when we come back to the issue of sustainability of agriculture. In large parts of the world, intense monoculture, especially of fruits and anything that requires pollination, requires bees. So if you don't work out, unless you're going to go around and brush pollen on by hand, which is not entirely sustainable, sustainable improvements in productivity, the sustainable part is really the crucial part. So creating an environment where bees can't live is not sustainable.
They've been sweeping up thousands of bees just down the east coast in Scotland. They're really, they can't find their hives.
No. Well, I mean, it's the same all over. I mean, California is the place where they have a real problem at the moment. Their entire fruit industry is under threat because of the lack of bees, and they can't even ship them in. So it is a major problem. It's also, I read a book not that long ago called The Rational Optimist, and it's a worthwhile read for anybody. Many of you have probably read it. Most predictions of doom, in fact, all predictions of doom, have generally been proved wrong, and they've largely been proved wrong because by the time they're predicted, the actions are already happening to deal with them. So, you know, look at the fish going back up the Thames, or any other example of a degree of cleaning up our act, recognizing what we've done and acting accordingly. So, you know, I'm relatively optimistic that we will find sustainable improvements in food production that will also address international food poverty.
Thank you.
Hi. I had a question slightly more on the note I was on last, just because we had a really interesting statistic in a lecture a while ago, and looking at your kind of cost-benefit analysis of everything, I was thinking, if 80% of the emerging species of infectious diseases come from animals, so they're zoonotic diseases, is it not a great risk having humans in such close contact to animals all the time by farming? Like, is that not a great risk?
It's a significant risk, and one of the ways in which production systems are changing, and one of the things we work on in Roslin, is to find solutions to zoonotic transmissions of diseases. So if we solve the problem, if we can find chickens that don't carry salmonella and find ways of assuring that that's not the case, it has a benefit to both the chickens and to us. But it is a significant source of infection. It's actually not necessarily the case that that the chickens give us the diseases, because of course the reverse is also true. And in fact, Staph aureus, one of the research that was done in Roslin a couple of years ago, showed that Staph aureus in chickens came from us. So it's two-way traffic, and it also, of course, goes into the wild. So we cheerfully transmit pathogens to all sorts of other species in the wild. We all live in the same world.
Okay, I think that's, I've been indicated that we have to close now, although it does say 8:30 on the website. Apparently 8 o'clock is the time we've got to stop. And I think finishing with the note of harmony in the world is a good one to stop with. I want to thank the audience for their questions, and I particularly want to thank David for being suitably provocative and opinionated and getting us all thinking. Thank you very much.
This production is brought to you by the University of Edinburgh.